My paper was gastric inhibitory polypeptide immunoneutralization attenuates development of obesity in mice. This paper was written by Michael O'Boylan, Patricia Glazebrook, and others, and this is presented by myself. Gastric inhibitory peptide is or otherwise known as GIP, is a 42 amino acid polypeptide synthesized in the enteroendocrine K cells in the intestinal mucosa of the duodenum and the proximal jejunum. The secretion of GIP is stimulated by nutrients such as glucose, fat, and hydrolyzed proteins. GIP binds to a G protein and stimulates the glucose absorption and inhibits lipolysis in adipocytes. It also increases nutrient uptake into bone and inhibits bone reabsorption. In the presence of glucose, GIP acts as a potent stimulator of insulin released by the pancreatic islet B cells. GIP accounts for 65 to 70% of postprandial insulin response. This response is your body's response to release insulin after you eat a meal. So after you eat a meal and your body starts digesting some of the food, your blood is full of has lots of glucose in it. So GIP helps signal that your body needs to uptake it, so it starts to increase the absorption of insulin. This also plays a role in type 2 diabetes, as it's shown in patients that the levels of GIP are diminished. It also shows the GIP half-life is decreased in circulating blood. It also decreases amount of meal stimulating. For this experiment, they used hybridoma cells and injected them with mouse GIP. They used the last 17 amino acids of the polypeptide, and they injected them into the mice, or into the hybridoma cells after 1 week, 4 weeks, 7 weeks, 10 weeks, and 2 weeks. They then screened for the antibodies using ELISA and identified which bind to the GIP in vitro by using a GIP receptor expressing cell line. These antibodies were then purified and were then ready to be injected into the mice for the diet study. For the diet study, the mice were injected with antibodies 5 times per week for 17 weeks. There was 3 different groups of mice. 2 groups were with high-fat diet, so their food was mainly composed of fat, and then the other was low-fat diet, composed mainly of carbohydrates and proteins. And each of these groups contained 10 mice each. And for the high-fat diet, there was 2 groups and one was administered antibodies specific to the GIP, and the other group was administered a control, which was PBS. For the results for this section, the high-fat diet mice, both groups gained roughly 10% of their body weight after the first week. They remained identical up to week 4, when the mice with the antibodies began to plateau while the control steadily increased. And then by week 7, the weights between the 2 groups became statistically significant. And then an overall picture of the whole diet study over the 17 weeks. The high-fat diet control gained an average of 21.5 grams, while the high-fat diet mice with the antibodies injected into them gained an average of 11.5 grams. This is a reduction of weight by 46.5%. In addition to all of these results, it showed that the control ate more food than the mice injected with antibodies. There was also no difference in the physical activity between each of the two high-fat mouse groups. It also, they also, this was seen by or shown by measuring their weight and amount of food after each Monday. The low-fat diet mice gained the same weight as the high-fat diet mediated mice, or with the mediated antibody mice, but they gained their weight at a much slower rate. This figure shows how rapidly the high-fat control group gained its weight and how slow the low-fat diet mice gained weight. It also shows how the mice injected with antibodies gained weight very rapidly in the beginning and then plateaued. The second part of this study was measuring how much fat each group of the mice with the high-fat diet accumulated over the 17 weeks. The results showed that the whole body fat was 2.3 folds higher in the high-fat diet control mice than it was in the high-fat diet mediated antibody mice. They also measured the amount of omental and subcutaneous fat within the mice, and the omental fat was 3.5 folds higher in the control, and the subcutaneous fat was also 2.2 folds higher in the control. In this figure, it shows the differences between the high-fat diet control and the high-fat diet with the mediated antibodies, and it just shows the percentage of what fat, or it shows the amount of fat accumulated in both studies. This picture shows a picture of the insides of the mice, and it shows the omentum fat, and as seen in the control, there is a lot more fat in the stomach, or in the insides of the control, than there is in the one injected with the antibodies. This is seen by all the white surrounding all the organs of the control, and then there is nearly nothing in the insides or there's nearly no fat surrounding the organs with the mice injected with the antibodies. My discussion questions were: do you think using mediated antibodies could be used to treat obesity in humans? And my second question is: GIP binds to a 7 transmembrane G protein receptors found on beta cells in the pancreas and is involved in the secretion of insulin. And what mechanism do you think it triggers the release of insulin?